Hello, today Nanagarn. Today we're going to be solving October-November 2023, Paper 1, Variant 2. And before we start, I'd highly appreciate it if you would take a moment out of your very busy day to subscribe to this channel and like this video. I'm currently offering free topic 1, 2, 3, and 4 notes. All you have to do is press on the website link in the description box down below. Question number 1. Which combination of lenses for a light microscope will give the greatest magnification? The combination which will give the greatest magnification is the highest number when you multiply both the eyepiece lens with the objective lens magnification. In this case, the answer is going to be D. Question number 2. The diagram shows an electron micrograph of virus particles in a human nucleus. What is the diameter of the labeled virus particle, which is this one? Now, prior, we've measured the actual image size on A4 paper, which was 3.5 millimeters. Now, we have to convert the millimeters to micrometers. We do this by multiplying the millimeters by 1000. And to convert to nanometers, we do this by multiplying by another 1000. Now, what is the diameter of the labeled virus particle? As we all know the magnification formula. Therefore, if you want to find out the diameter of the labeled virus particle, which is the actual size, you have to divide the image size by the magnification. Therefore, we're going to divide this number here by the magnification. Therefore, this is going to give us this number right here. Therefore, this is equivalent to D, if we were to assume that we rounded up. Question number 3. What is correct about the synthesis and release of glycoprotein mucin in goblet cells? So as we all know, proteins are formed on the rough endoplasmic reticulum, which contain ribosomes on top. And now, these proteins are further modified by moving to the Golgi body via being packaged into secretory vesicles and then being modified in the Golgi body. In this case, it's a glycoprotein, so a carbohydrate group is added on the Golgi body. Now, let's see the suggestions that we have here. 1. The protein is produced on ribosomes, and a carbohydrate chain is added in the Golgi body. This is correct. Now for 2, the glycoproteins are packaged into vesicles in the Golgi body, forming lysosomes. Now, this is completely incorrect. The reason for this is that lysosomes are formed by packaging hydrolytic enzymes, not glycoproteins. Therefore, this is incorrect. For 3, secretory vesicles containing the glycoprotein move from the Golgi body and fuse with the cell surface membrane. Yes, this is the process of exocytosis. After the protein is further modified on the Golgi body, they are again packaged into secretory vesicles and then move via the cytoskeleton to the cell surface membrane for exocytosis. Therefore, the answer is going to be B. Question number 4. What are the functions of microtubules? So, functions of microtubules consist of forming the cell shape and the transport system of the cell, or creating the cytoskeleton, as well as forming the spindle fiber. Now, let's see the suggestions that we have here. 1. Aligned movement of cilia in the bronchus. So let's break this down. The cilia structure consists of 2 central microtubules and then 9 microtubule doublets, a ring of 9 microtubule doublets. Therefore, they are formed of microtubules. Therefore, this is correct. 2. Attachment to centromeres during metaphase. So, as I just mentioned, the functions of microtubules is also forming the spindle fibers. Now, what happens during metaphase is that, let's draw this down. The centrosomes are the structure that creates the spindle or forms the spindle. And now the spindle attaches to the centromere or the kinetochore, pulling, so from both sides, pulling the chromosome apart. So, pulling it apart. Therefore, this is correct. 3. Moving secretory vesicles around a cell. So we just mentioned that microtubules form the transport system. Therefore, they do move secretory vesicles, and the answer is going to be A. Question number 5. Which organelles contain nucleic acids? So, Golgi body does not contain nucleic acid. The function of Golgi body is after the protein has been formed on the rough endoplasmic reticulum, they are further modified or released from the rough endoplasmic reticulum via secretory vesicles and then further modified at the Golgi body, where, for an example, a carbohydrate is added. Now, this means that it does not contain nucleic acids. Now, here we have mitochondria and chloroplasts. So, mitochondria contains its own circular DNA, as well as the chloroplasts. They don't contain the DNA, which is the double helix. They contain a smaller circular DNA, or as we say, a prokaryotic DNA. Therefore, both of these are correct, and the answer is going to be D. Question number 6. Some features of cells are listed. Which features can be found in plant cells and in prokaryotic cells? So, for cell walls, both plant cells and prokaryotic cells have a cell wall. Plant cells is formed of cellulose and prokaryotic cells is formed of murine. For 2, cell surface membrane. Yes, both of them do have a cell surface membrane. And ribosomes. Yes, both of them do have ribosomes. Therefore, the answer is going to be A. Number 7, which bonds are present in all viruses? So as we all know, all viruses must contain a nucleic acid, whether it's a DNA or an RNA, but be careful, it's not both at the same time. It's either a DNA or an RNA virus, and must contain a capsid or a protein capsid. Sometimes viruses do contain an envelope, but here it's not in all viruses. Therefore, as we mentioned, a nucleic acid and a capsid. Now, here we have the options: phosphodiester, peptide, and covalent bonds. Phosphodiester bond is a chemical bond formed with the phosphate group and the base. Therefore, this forms the nucleic acid backbone. Therefore, this is correct. Now for 2, peptide. As we just mentioned, all viruses must contain a capsid, and we know a capsid is made of capsomeres. Capsomeres are made of proteins, therefore it must have a peptide bond. Now the third option is covalent. So both phosphodiester and peptide bonds are covalent bonds. Therefore these are correct because a peptide bond and phosphodiester bond are strong covalent bound. Therefore the answer is going to be A. Question number 8. A student carried out Benedict's test on 4 different concentrations of glucose solution and then recorded the time taken for the first appearance of color change. The student found it difficult to identify the first appearance of a color change and consistently timed each solution for 2 seconds after the color change first appeared. This introduced a source of error into the experiment. Which statement about this error are correct? Now, first of all, Benedict's test is a semi-quantitative test used to test for the presence of reducing sugars in a solution. With semi-quantitative, this means that the rate of color change and the color change itself could approximately determine the reducing sugar concentration in the solution. Now, the type of error here is called a systematic error. Now, let's define a systematic error. For example, if a balance is miscalibrated or not calibrated properly at the start of an experiment, this would introduce a systematic error. Therefore, any substance that is weighed would be 0.5 kilograms extra at the start. This is an example. Therefore, this is called a systematic error. Versus a random error. A random error happens when there is an alteration in the environment. So in this case, if the room temperature suddenly dropped or risen, this would introduce a random error, and this can be resolved by repetition. Now, which statements about this error are correct? 1. The effect of the error would be reduced if the student performs 3 repeats at each concentration of glucose. So, as we just mentioned, because this is a systematic error, aka an error in the calibration, which is the student timing the solution for 2 seconds after the color change, this would not be reduced by repeating. Therefore, this is incorrect. This would be reduced in this case if this were a random error. Now for 2, the error will prevent the student from identifying which solution has the highest concentration of glucose. So this is incorrect because, as I just mentioned, Benedict's test is a semi-quantitative test. This means that, for example, if the color change happens after 2 seconds and then in the other solution the color change happens after 6 seconds, this means that the first solution has a higher concentration of reducing sugars. So even if the student were to time the solution for 2 seconds after the color change, the rate of color change itself will determine approximately the concentration of reducing sugar, or which solution has the highest or the lowest concentration of glucose. Therefore, this is incorrect. 3. The error is systematic, as the student consistently timed each solution for 2 seconds after the endpoint. This is what we just mentioned. Therefore, only 3 is correct. Therefore, the answer is going to be D. Question number 9. Which statements about amylopectin and glycogen are correct? So, both are polysaccharides. And the difference between amylopectin and glycogen is that amylopectin is branched every 20 monomers. This is for amylopectin. And glycogen is branched every 10 monomers. Therefore, glycogen is more branched or has more branches. Now let's see the suggestions that we have here. 1. Amylopectin and glycogen contain 1 to 4 glycosidic bonds. Yes, this is correct. They both contain 1 to 4 and 1 to 6 glycosidic bonds to form the branches. 2. Amylopectin contains beta-glucose. This is incorrect. Both of them contain alpha-glucose monomers. 3. Glycogen contains more 1 to 6 branches than amylopectin. I just mentioned the 1 to 6 bonds are formed within the branches. And as I also mentioned, glycogen forms branches every 10 monomers. Therefore, it's going to be more branched. Therefore, has more 1 to 6 glycosidic bonds. Therefore, this is correct. And the answer is going to be B. Question number 10. How many statements about fatty acids are correct? Let's see the suggestions that we have here. Fatty acids are either saturated or unsaturated. This is correct. If it is a saturated fatty acid, it does not contain any double bonds. And if it's unsaturated, it must contain at least 1 double bond. Now, the second point, fatty acids always have at least 2 double bonds. Now, this is incorrect. It has at least 1 double bond because let's assume this is a long hydrocarbon chain. The end of the chain contains this formula here, a carboxyl group. Therefore, has a C carbon to oxygen double bond, so must contain at least 1 double bond. But I cannot assume that this fatty acid is saturated or unsaturated. I would never know whether it's saturated or unsaturated. Therefore, this cannot be correct in the situation, because if we were to assume that this was a saturated fatty acid, then it would only contain 1, so this is incorrect. Now, for 3, long chains of fatty acids are very good energy stores. So, let's assume that a fatty acid had a very long hydrocarbon chain. Breaking these bonds releases energy. So the more bonds broken, the more energy released. Therefore, longer chains of fatty acids are better energy stores than the shorter ones. The fourth point, fatty acids are insoluble in water and do not affect the water potential. So, a rule here, if a substance does not dissolve, it does not alter the water potential because it does not affect the osmolarity in any way. Therefore, this is correct. So we have 3 points here, and the answer is going to be C. Question number 11. The diagram represents an amino acid or represents a variable side chain. What is not a possible side chain? So at your current level, you're not expected to know what these side chains correspond to which proteins. How to solve this question indeed is very simple. You have to draw out each formula in this case. So let's assume that we drew all the formulas out, and then we have the last suggestion here. So the only way to solve this is that you have to have a prior knowledge that carbon forms 4 covalent bonds, or in this case looks like this. forms 4 covalent bonds with the hydrogen. So in order for a side chain to effectively bind, or anyways to bind to the carbon, there must be one free bond in order to bind with the carbon here. For as an example, we could see this in the first, because there is a free bond which is able to bind to the side chain. But in this last option that we have here, there are no free bonds at all. Therefore, this is not a possible side chain. Therefore, the answer is going to be D. Question number 12. The graph shows the effect of an enzyme on a reaction. Which row identifies X, Y, and Z? So, looking at this diagram, you must identify first that this is an exothermic reaction. The reason for this is that there is a lower energy level at the product than what it started. Therefore, energy is released here, just for your information. Now, let's see the suggestions that we have here. X, Y, and Z. We either have catalyzed, uncatalyzed, energy lost, or energy gained. So, as we all know, enzymes are biological catalysts. Therefore, they lower the activation energy needed in a system. Therefore, it makes sense that Y is the catalyzed solution with an enzyme, because there is a lower overall activation energy. And therefore, X is the uncatalyzed. And then Z, as we mentioned earlier, is the energy lost by a system, or the energy released during a reaction. So energy loss is same as energy released. This would be an endothermic reaction, or energy gained by product, if the overall energy of the system was higher at the end. Therefore, the answer is going to be D. Question number 13. The graph shows the results of 2 experiments on the effect of increasing substrate concentration on the rate of enzyme catalyzed reaction. One experiment was at high concentration of enzyme, and the second was at low concentration of enzyme. All other variables were standardized. What would limit the initial rate of reaction at point X? So here we have the initial rate of reaction, and here we have the substrate concentration. And here we have high enzyme concentration and low enzyme concentration. So at this point right here, the substrate concentration is constant. Therefore, the only option here that would limit the initial rate of reaction is the enzyme concentration, because a limiting factor, what is a limiting factor? Is because if I added a substance, it would increase the overall rate of the reaction. Therefore, in this case, when I added more enzyme concentration, suddenly the rate of reaction increased. Therefore, this means that the enzyme concentration was the limiting factor in this case, since the substrate concentration is constant here. Therefore, the answer is going to be A. Question number 14. Which statements about Michaelis-Menten constant of an enzyme are correct? So, Michaelis-Menten constant, or Km, is the substrate concentration where an enzyme operates at half of its Vmax, or maximum rate. And Michaelis-Menten constant is a measure of the efficiency of the enzyme, because the lower the Km, the higher the efficiency of the enzyme, and vice versa. The reason for this is, let's say, the lower the Km, the less substrate concentration is needed to reach half Vmax. So let's see the suggestions that we have here. For one, at the Km value, half the active sites of the enzyme should be occupied by the substrate. So this is correct, because as we said, the definition of Km is the substrate concentration where an enzyme acts at its half of its Vmax. Therefore, half of the active sites of the enzyme should be occupied, because it's half Vmax. Therefore, this is correct. For 2, Km represents the substrate concentration at which enzyme is working at half of its maximum rate. So we just mentioned this is correct. Now, for the third option here, the lower the Michaelis-Menten constant, the lower the affinity of the enzyme. We said this is incorrect. The higher the affinity, the less substrate it needs to reach its Vmax, half its Vmax. And for option 4, when an enzyme has a high Km value, the enzyme catalyzed reaction will proceed very slowly. Yes, this is correct because we just mentioned about the efficiency of the enzyme. The higher the Km, the less efficient the enzyme. Therefore, the answer is going to be B. Question number 15. Which component of cell surface membranes help to reduce fluidity of the phospholipids at high temperatures? So, reduce fluidity at high temperatures. Let's see the suggestions that we have here. A. Phosphate groups. This is not the case here. For B. Unsaturated fatty acids. So, this is actually incorrect because it increases the fluidity. The reason for this is that unsaturated acids, fatty acids, contain carbon-to-carbon double bonds. These carbon-to-carbon double bonds create, let's say, gaps within the membrane. These gaps increase the fluidity. Therefore, this is incorrect for C, cholesterol. Yes, this is correct because at high temperatures, cholesterol works to reduce the fluidity, and at low temperatures, cholesterol works to increase the fluidity. Therefore, this is correct. And then finally, proteins. This is incorrect. Therefore, the answer is going to be C. Question number 16. Plant cells were left for 50 minutes in 3 different sugar solutions. The water potential in the cytoplasm of 3 cells were at the same at the start of the experiment. The diagram shows the appearance of these cells after 50 minutes using a light microscope. So here we have cell P, cell Q, cell R. Cell Q clearly looks turgid, and cell R is plasmolysed. Be careful, this is not flaccid, it's plasmolysed because it's starting to pull apart, so the cell surface membrane pulled apart from the cell wall, so in this case it's plasmolysed. It would be flaccid if the cell membrane was still attached to the cell wall. Now, which conclusion is correct? Let's see the suggestions that we have here. A. Cell P has the same concentration of sugar inside and outside the cell. So, in this case here, we are talking about water potential, not osmolarity. Therefore, we would say that it has the same water potential, not the same concentration of sugar inside. Therefore, this is incorrect. We must here mention water potential. For B, cell Q is flaccid and cell P is plasmolysed. So, this is incorrect. Cell P is not plasmolysed. Cell R is plasmolysed. This is incorrect. And cell Q is not flaccid, it's turgid. For C, cell Q was placed in 1% solution. So 1% solution is the lowest concentration of sugar. Therefore, it has the highest water potential because here it's the lowest concentration out of all. It has the highest water potential. As we all know, water potential moves from a higher concentration gradient down to a lower concentration gradient. Therefore, the cell became turgid. This only indicates that cell Q was placed in the solution with the highest water potential. Therefore, this is correct. For D, the sugar solution outside R has a less negative water potential. Less negative water potential here means a higher water potential. This is incorrect. It had a lower water potential because water moves from the higher to lower water potential. Therefore, it moved out, not in, indicating that the cell itself here had a higher water potential and the solution had the lowest water potential, therefore the highest concentration of glucose, here 10%. Therefore, this is incorrect. It should be a more negative water potential. Therefore, the answer is going to be C. Question number 17. The graph shows the effect of increasing the side length of agar cubes on the surface area and the volume of the cubes. Which row correctly identifies line 1, 2, and the effect of increasing side length on the surface area to volume ratio? So, let's assume that we have here a cube which has sides of 2 centimeters and another cube with a side of 4 centimeters. Now, let's calculate the volume, surface area, and surface area to volume ratio for each. Now, for the volume, it's going to be 2 times 2 times by 2 is going to be 8 centimeters cubed. And then let's calculate the surface area. In this case, we times 2 by 2 and then times this answer by 6, because a cube has 6 sides, is going to be 24. And the reason why I'm calculating this, because you guys requested before to do the actual calculations. Therefore, this is why I'm doing it. And therefore, the surface area to volume ratio in this case is going to be 3:1. Now let's do the same thing for the volume. For the second cube, it's going to be 64. So 4 times by 4 times by 4, it's going to be 64. The surface area in this case is 4 times by 4, then times by 6, which is 96. Now here the surface area to volume ratio is going to be 96 divided by 64. So 1.5 to 1. So we could clearly see the first conclusion is that the surface area to volume ratio decreases as we increase the side length. So it decreases. If we were to compare the volume with the surface area, we can see here that the volume is 8 and then it's 24. In proportion to the volume, as the length increases, it's now 64 to 96. So as we could see, the volume seems to be increasing at a higher proportion. So obviously this is all an estimate. We can conclude that the volume is increasing at a higher proportion than the surface area, because it's almost now near to 96 in contrast to 8 and 24. Therefore, it makes sense that here, or the accelerating line, is going to be the volume. Therefore, line 1 is volume. And therefore, line 2, because it's increasing at a lower proportion, is going to be the surface area. Therefore, the answer is going to be C. Question number 18. A human embryo consists of 12 cells at the start of day 1. The cells divide by mitosis for 4 complete days at a rate of 1 division every 32 hours. What is the maximum number of cells in the embryo at the end of day 4? So a day has 24 hours. So what we're going to do is multiply 4, because it's 4 days, by 24. This is going to give us 96 hours. Now what we're going to do is, because a rate of 1 division every 32 hours, so we want to know how many divisions within this time. Therefore, 96 divided by 32, in this case, is going to be 3 divisions. Now, the way mitosis works is, for example, if we start at 12 cells, the first division, it doubles to 24. And then in the second division, it doubles to 48. And then in the third division, it doubles to 96. Therefore, the final number of cells after 3 consecutive divisions is going to be 96 cells. Therefore, the answer is going to be B. Number 19, which diagrams show roles of mitosis? So, mitosis is the dividing of cells into 2 identical daughter cells, and roles of mitosis are in growth. Meiosis happens in the gametes itself. Now, let's see the suggestions that we have here. Here, in all the options, we could clearly see signs of growth. In the plant, and in the second option here, we could see growth. In the third option, there's growth of the roots and the leaves. And in 4, we could see the growth of one arm. Any growth happens via mitosis, therefore all of them are correct, and the answer is going to be A. Question number 20, which events are part of the mitotic cell cycle? So let's draw the entire cell cycle. We have mitosis, which consists of prophase, metaphase, anaphase, and telophase, and then we have the G1 phase, the S phase, which is slightly smaller, where DNA replication takes place, and then the G2 phase, where the cell continues to grow after DNA replication. Now, let's see the suggestions that we have here. Which events are part of the mitotic cell cycle? We have interphase, anaphase, cytokinesis. So, interphase is what we call in everything else other than the mitosis itself. So, G1, S, and G2 is part of the interphase, therefore this is correct. For 2, anaphase. So, part of the mitosis is anaphase, and this is correct. This is where the chromosomes are separated apart. Now, for 3, cytokinesis. So cytokinesis happens directly at the end of mitosis, where the actual cell finally divides into 2 identical cells. Therefore, this is correct, and the answer is going to be A. Thank you very much for watching this video. Stay tuned for part 2, which will be released very soon, and don't forget to subscribe to this channel and like this video.